Despite recent advances, patients with high-risk acute myeloid leukemia still face poor outcomes. CD123 and CD33 CAR T cells have so far shown limited efficacy and some toxicities, which highlights the need for new constructs and alternative targets. So we hypothesized that we could have a safer and more efficient new CAR by using NK cell, by using IL-12, and by using new targets. So why NK cell? NK cell obviously have unique properties to kill tumor cells, but when they are activated by cytokines, they have more proliferation, more survival, and more potential to kill tumor cells. Also, compared to CAR T cell, CAR NK cell have shown a safer safety profile, a better safety profile. And it can also be delivered off the shelf. And for all those reasons, there is clinical trials that are being developed showing promising results. So now why IL-12? IL-12 has unique potential to enhance NK cell proliferation and cytotoxic potential. IL-12, compared to other cytokines, has a broader immunomodulatory effects on various immune cells. It can bridge innate and adaptive immune responses. And finally, why new targets? So we identified mesothelin and CD70 as promising targets. They are expressed on several tumors, but on AML, mesothelin is expressed in 36% of AML, and CD70 is expressed in up to 86% of AML. So the reason for choosing them is also because they are absent on the hematopoietic stem cells and enrich in leukemia stem cells. So this is crucial because compared to CD23 or CD123, they can reduce, targeting them will reduce the risk of myeloablation. So with that in mind, we leverage these advantages to build new constructs, a mesothelin CAR NK cell secreting IL-12. First, we have shown that this CAR NK secreting IL-12 and targeting mesothelin has a benign secretome. When we compare it to CAR T secreting IL-12, we saw lower level of inflammatory cytokines. Then we compared this construct with the same construct but secreting IL-15. So it's CAR NK, one secreting IL-12, the other one secreting IL-15. And what we see, which is very interesting, that with IL-12 we have more production of interferon gamma, TNF alpha, granzyme B, and also in a re-challenge, we see at long term better tumor control in an ovarian model. So building upon this, we use this mesothelin CAR NK construct secreting IL-12, and we show that in vitro it has a high cytotoxic potential against number one AML cell line secreting expressing mesothelin. So it has better, higher cytotoxicity than mesothelin CAR NK without IL-12 and than NK cell. Then we did an in vivo model with IL-15 NSG mice, and we show that we have a better tumor control with this IL-12 mesothelin CAR NK cell compared to mesothelin CAR without IL-12. Then we did similar experiments, but with another construct, except of targeting mesothelin, we target CD70. And we show in 5 different AML cell lines that the CD70 CAR secreting IL-12 has a higher cytotoxicity against those cell lines, irrespective of TP53 mutation status. We also show higher interferon gamma production. And finally, which is interesting, we show the ability of this IL-12 secreting CAR to activate bystander NK cell, which secretes more interferon gamma and have more degranulation, and thus are more able also to help killing the tumor. So finally, what we see with this study that it is feasible and safe to engineer IL-12 secreting CAR NK cell. We have now promising results for using this CAR. At this stage, I think we now have the rationale to build a clinical trial with this construct, and it's a promising, possible off-the-shelf approach for the patient. It has also maybe a broad therapeutic potential because CD70 and mesothelin is expressed on other cancers such as hematopoietic cancer and also a solid tumor. We already have good results with this construct in ovarian cancer and renal cell carcinoma, and we're working on the lymphoma model. I just would like to thank everybody in the Rémy lab, because without them, this study will not be possible, and I'm especially thankful to Dr. Rémy, Fugo, Liu, Jiung Zhang, and Mubin Taranum.